Thank you, Lakmal, for the introduction here. It's been a wonderful 2 days at this conference here, and it's been super exciting to see all the excitement for protein sequencing, and it really helps motivate us developing this technology. And it makes sense because, as it's been said, that proteins are really the functional ingredients or the active ingredients in life. I like to think about them. The rest of this stuff is just sort of it's like carbs. It's just like filler ingredients there. So proteins. So Lakmal gave a good introduction yesterday to some of the motivations of proteomics and what makes proteomics analysis so challenging. So we know that proteins are composed of 20 strings of 20 amino acids, right? And what makes them very, very interesting and gives them such functionality and dynamics to them is what's called post-translational modifications, right? These are modifications that happen at the translational level, and I like to think of them like real-time point mutations that happen to proteins that allows how cells are able to function and respond to things in real time is largely governed by PTMs and these different signaling pathways that are regulated by PTMs. And so what this means is you might have one protein sequence, right? Essentially one primary sequence, and that's encoded in the genome. But for that single protein sequence, when it's actually expressed and translated, there's potentially many unique variations of that protein. They can be present in different copy numbers, and each of those can have a completely different function from the other ones. So just to give you an example here, so this is everyone's favorite oncoprotein P53, and this is just a small subset of known variants, PTM variants of P53. And what the field has adopted, different PTM variants, is the term proteoforms. So these are 13 or a little bit less of the known P53 proteoforms. And what you'll see here is that they vary, right, in not just the types of modifications, but also in different locations and as well as the combinations. So some are shared, some are unique across them. And the importance of this is that each of these different proteoforms can have a completely different function from the other one. Okay? And that means each of them can then represent either a novel biomarker or drug target. And the only way that you would be able, right, to resolve this is by looking at the full length of these protein molecules to really resolve these combinatorics that exist at the protein level, right? So just like long-read sequencing is important for DNA and RNA, as you all know, it's also incredibly important for proteomics as well. Okay. So it was talked about yesterday. This diversity, molecular diversity, really explodes at the level of the proteome, right? So you have over 1 million different protein variants, largely because of these PTMs, in addition to splice variants. And if we look, and this is an incredible analytical challenge, right? And so if we look at the sequencing technologies that we have available to us at each of these different levels, as we know, sequencing at scale does not exist for proteins like it does for DNA and RNA. And so who do we blame for that? I blame Francis Crick for that because he came up with something called the central dogma, okay? And he wrote about this first in this paper from the 1950s here, in which he described the information flow in biological systems. And there's one passage in particular that I'd like to highlight. It says, once information has gotten into a protein, it can't get out again. Okay? And he drew these nice diagrams here describing this information flow, DNA to RNA to proteins. You can go back from RNA back to DNA. You can make copies of RNA, copies of DNA. But you can never have this. Right? You can never have proteins you can never make copies of a protein, nor can you back translate protein sequences back into the sequences that coded for them. And so this is sort of the fundamental challenge then of protein sequencing, is that if you think about all the sequencing technologies that exist today, they're all relying on some form of either replication or some ability to copy that molecule, right, for DNA and RNA. Except for one technology, of course, which you probably can guess. So if we look at current technologies, and I'm just going to pick on one here. So if you have your sample, your proteomic sample, one of the most common ways to analyze that is protein mass spectrometry, in particular bottom-up mass spectrometry. And it's incredibly powerful technology. You can get great depth and great coverage width as far as protein identification, but what you end up with is very limited sequence coverage. So though you might be able to identify a lot of proteins within your sample, you're actually identifying them by just recognizing very, very short fragments of that protein, and your average protein that you ID, you've only ID'd based on covering about 30% of that protein's overall sequence. Right? So you're missing a lot of information, which means you don't have the ability to reconstruct full-length protein molecules. In addition to that, you only find what you're looking for, because it's largely a reference-based technology. It's complex and expensive instrumentation. Are there any mass spectrometrists in the audience right now? No? OK, so we can speak freely. Nobody likes mass spectrometry, right? OK. I'm just kidding. Some of my best friends are mass spectrometrists, actually. OK, so the idea here is that nanopores could potentially complement existing proteomic technologies by offering more complete coverage, being single molecule, so inherently quantitative, and you'd be able to get that full-length, potentially full-length information, which is what my research is really focused on. So what are the challenges then with threading proteins through nanopores? So what's nice about DNA and RNA, they have this nice negatively charged background, which is nice when you're looking at them through these electrical nanopore sensors. You have some level of control just based on that charge. Proteins, on the other hand, are heterogeneously charged. They also, if you're looking at full-length proteins, they fold up into these stable tertiary structures, right? Which you need to have some way of unraveling them and processing them linearly through a nanopore. OK. So I did my PhD work in Mark Akeson's lab, who developed some of the fundamental breakthroughs for DNA-based sequencing using nanopores. And it was around the same time that I was doing my PhD that these two papers came out, which really showed, in my view, what the secret sauce was to enable DNA-based protein sequencing. And that's the combination of motor control along with a pore that is sensitive to single nucleotides. Okay? It's able to achieve single nucleotide resolution. And so seeing sort of the breakthroughs that happened and coming with a background in protein biochemistry, I thought the next thing to do then is to try and enable nanopore-based protein sequencing. And so inspired by this work, I knew the most important thing that we would need to do protein-based nanopore sequencing was a motor to control the translocation of full-length proteins through the nanopore. So if we look into nature, protein translocation across membranes and into different compartments is a pretty ubiquitous process. So there's a lot of different molecular motors, molecular mechanisms that nature has evolved to transport folded proteins in this way. And so one of these molecular motors in particular that we work with is called ClipX, and it's known as a protein unfoldase translocase. And the way that it works inside of a cell is that ClipX recognizes specifically tagged proteins at the C terminal. It grabs onto that tag and it starts pulling on this protein until it's able to destabilize it, unfold it, and then it translocates it into another protein complex called ClipP. And ClipP, you can think about it like this barrel protease, and it chops up that targeted protein for degradation. Okay? So ClipXP, sort of what this complex is known as. And in the cell, it's to do this targeted degradation. And so over 10 years ago, we showed published the first papers in which we showed that we could use this ClipX, either by itself or in complex with ClipP, to enable process of translocation of very long protein strands through a nanopore. Okay? And in this particular setup, what we have here is we're showing a model protein substrate. It's consisting of several different protein domains that we've engineered essentially as one fusion protein. And then on the C-terminal of that, we have this anionic peptide sequence in addition to the SSRA tag, and that's what ClipX recognizes. And what happens in this process is when you load that protein into the flow cell, onto the top of the flow cell, basically the cis side, it electrophoretically threads into that pore, and it gets stuck because of these stable folded domains. And then in this setup, we have the ClipX motors on the trans side. So it grabs that, the SSRA tag, and then it catalyzes unfolding and processive translocation of that entire protein strand through the pore. And it generates signals like this. So this is showing you a single trace from this model protein here. And the pore that we're using at this time, we're just using these single channel nanopore setups. We weren't using minions or anything like that. And the pore we're using, called alpha hemolysin, it's a very sort of low resolution pore. It's not sensitive enough to the structural dimensions don't make it sensitive to single amino acids, is what we found out, and it was known for a long time. It also was not suitable for DNA sequencing either. But what we were able to get from this analysis is sort of a coarse-grained view of this protein's, mainly its tertiary structures, in which we can identify pre-unfolding states and translocation states associated with each of these protein domains as they were pulled through the pore by ClipX. And just based on this coarse-grained analysis, we had some ability to resolve between some sequence-dependent features just by looking at the overall signatures of these different domains. We also got information about the stability of individual domains based on how long they take to unfold, and also the length of these domains based on how long they take to translocate. Okay, so fast forwarding now, we published that over a decade now ago, and so fast forwarding to present day, a few years ago and starting at my own lab, wanted to revisit this protein sequencing approach. This challenge still hadn't been solved, and so going back to trying to recreate the secret sauce, we liked the motor that we were using previously, but now we wanted to see, now that the nanopore technology had advanced quite a bit, what we could do with a more sensitive nanopore setup. And for that, we started collaborating with Oxford Nanopore Technologies to see if we can get this ClipX unfold-A system working on their MinION, right? And just using their commercially available CSGG R94 pore arrays. So all the data that I'm going to be showing you here is just from the off-the-shelf R94 pores. All right, now the trick here to get this to work, to get the ClipX setup to work on these commercially available devices, if you remember, we had the motor in the trans side, basically underneath the pore in our previous setup. And so with the commercially off-the-shelf devices here, we couldn't do that. We couldn't put the motor into that trans side solution. And for a lot of technical reasons, manufacturing flow cells that have an enzyme present in the trans is quite challenging. So what we wanted to do first is come up with a cis side solution to implementing this unfold-based technique. So what we have here is somewhat similar to what I showed you previously. We have an analyte protein now in blue. Okay. At the N terminal, we attach a very negatively charged long peptide, polypeptide, and at the C terminal, we have what we call a blocking domain and this SSRA tag there. Now, what happens when you load this protein into the flow cell, you can get electrophoretic threading of that strand into the pore. It gets stuck because of this blocking domain. Then we introduce ClipX into solution, and then it pulls that protein back up out of the pore. Okay? And it's this part of the process, when that motor's pulling that strand back up through the pore, what we found in these R94 pores is that really these quite complex, intricate signals with much, much better resolution than what we were seeing previously using alpha hemolysin, okay, which got us quite excited now. So to sort of explore this, the sensitivity we had to single amino acids, what we did, we engineered these libraries of different synthetic protein strands in which we systematically made different substitutions within the regions of this strand in order for us to sort of start to begin to understand how sequence determines the signal levels that we observe. Right? So I'm showing you one example strand here where you can see the different signal levels that are attributed to just a single point mutation within this long protein strand. So each of these blue regions here is around 29 amino acid linker, primarily glycine serine, and then we introduce at these different locations a different point mutation. You can see, depending on that mutation, it can manifest very large deviations in the ionic current signal. So the other cool thing that we can see with this approach is if you zoom in really closely, what we can begin to resolve is individual steps within that signal, okay, which correspond to actually individual steps that ClipX is making as it translocates that pore through the as it translocates that protein strand through the pore. And using this data, we're able to essentially make the highest resolution measurements to date on the activity, the functional activity of this motor, and we determine it takes the average step size of about 2 amino acids, which is not bad for a sequencing motor, considering that your Kmer size, the amount of amino acids that are contributing to your ionic current signal within the pore, is actually much larger than that. And then, so the next thing that we did to sort of determine with what sensitivity we can classify amongst these different substitutions that we made within these strands, we're able to do a bunch of signal processing. We extract, we label our raw signal data, and then we feed it into machine learning classifiers, just using a supervised approach to get an idea of how sensitive this method is now for classifying against these different amino acids. And these are the results that we have here. And so if you're looking at the top N, this is a plot of the top N accuracy for 20-way amino acid classification. If we look at the so the top one here, we get an accuracy of around 30%. So this would be pretty bad if this was DNA sequencing, right? It would essentially be random, but remind you this is 20-way classification problem now, so it's well above essentially the dummy baseline of 5%. Of course, we would like to, we're trying to figure out ways to improve this accuracy. So we know that if you could read that same molecule again, that would give you a great way to essentially average out the noise that you observe across any single read and be able to come up with a single molecule consensus. And so, sort of by accident, what we found out is that we were able to develop essentially like a single molecule nanopore flossing technique that allowed us to reread the same protein molecule multiple times. And this is achieved by having what we found out is a particular amino acid motif that ClipX slips on. And when you put this motif at the very end of your protein, in that adapter that goes on the N terminal, when ClipX comes up to the end of the protein, it hits that sequence that actually loses grip of the protein, and then the protein slips back down into the pore, and at some point later, ClipX can re-engage, and then it restarts reading. Okay, so you're seeing 3 near-complete rereads of this particular protein molecule through the pore. So using a system like this on these model proteins, we're able to achieve around an average of 10 rereads per molecule. And what that buys us in this 20-way classification problem is going from 30% with a single read up to 60% substitution calling accuracy in that 20-way classification task with that 10 reads. All right. So I started off with a big motivation and talking about the importance of proteoforms and PTMs, right? So the next question that we wanted to ask is, what is the sensitivity to this method to post-translational modifications, in phosphorylation in particular, as it being one of the most critical known PTMs in protein biology. So I'm showing you here on the bottom left, this is a pile-up of many traces that have been aligned in time using dynamic time warping. In orange here, there's no phosphorylation on any of these molecules. And then we take the same protein sample and we mix it with some a cocktail of different kinases that recognizes specific motifs within that protein strand, and then we put it back onto the nanopore, and then these are the types of signals we can now observe. So this is a pileup of different traces in which you can see now large deviations compared to that background reference trace, where you can see at specific sites that correspond to these phosphorylation sites. So we can see differences between no phosphorylation versus a single phosphorylation versus 2 phosphorylations that are co-localized on this strand. And so the cool thing about this is this protein strand is a few hundred amino acids in length. It has many possible phosphorylation sites on it. So that means there's many possible combinations in which they occur on any given molecule. And from a single sample, what we were able to observe following this assay, we were able to quantify over 100 unique proteoforms of this model protein from just that single sample. So again, model protein, right? So it's a very synthetic sequence that we're using here as a foundation on which to build on, but super exciting in that this is the only a technology that would enable you to resolve proteoforms at this level of resolution over this length of protein molecule. And we think that's super exciting as far as being able to apply that to natural proteins. All right. So how about folded proteins? So a lot of these synthetic ones I was showing you had little to no structure. So folded proteins are a little bit trickier, right? So the way that we deal with those, they essentially have to be unfolded and transported across that pore twice, right? So the first way we do it is electrophoretically, using the supercharged linker that essentially, when it initially threads into that pore, it's able to pull on that protein with enough force, tens of piconewtons of force, such that it can, for many domains, mechanically destabilize them. And then when you pull enough of that protein down through into that trans chamber, what can happen is actually the protein can start to refold. Right? And refolding can actually catalyze translocation of that entire protein domain through the pore. And this is a process that's known to happen in nature and serves as one mechanism of naturally translocating proteins across membranes. It's known as co-translocational folding. And we can rely on that to help pull these larger structures through the pore. Then after we're able to do that, ClipX is able to work as it did before and process these protein strands back up through the pore. So I'm just showing you example traces here of now much more complex signals owing to these folded domain sequences. So examples from variants of titanide 27 domain as well as amyloid beta here. Where we're getting these much more complex signals that are now much more challenging for us to map back what particular regions of the sequence does this signal correspond to. So as going towards trying to solve this challenge, what we're really focused on right now is building protein sequence to squiggle models. Okay? And based on the data that we have right now is that we see a very good correlation between the volume of the protein sequence and charge of that protein sequence correlates very well to the signal levels that we observe. And so right now we're able to make these, albeit I would say rudimentary at this time, protein sequence to squiggle models that actually still, though fairly simple, they're just based on the biophysical properties of that protein sequence in a sliding window. We're able to pretty accurately predict or model what the raw signals of our synthetic protein strands actually look like. So in the black here is a real signal compared to what our model predicts that signal to be, just based on the sequence of it. And so the reason that we're really interested in building these models is because we think it could enable essentially a full-length proteoform fingerprinting approach, what we call essentially squiggle matching. So you can imagine it like this. You would have a reference sequence database. So take a reference human proteome, something like that, a bunch of different protein sequences of interest. You would put all of those sequences through your sequence to squiggle model. You generate a reference database of now squiggles, a squiggle database. And then you could take your unknown protein sample, generate a bunch of squiggles, unknown squiggles, or squiggles manifesting from unknown proteins. And the way that you can ID them is essentially now you can do squiggle-based matching, where you use techniques such as dynamic time warping, where you can essentially calculate a similarity between your unknown squiggle and the squiggles in your simulated database. And essentially that would enable a nearest neighbor squiggle classification approach just based on what squiggle it was your squiggle most similar to, right? And the cool thing about this is that you also have the potential, because it's a biophysical model here, to model in what potential PTMs, what their squiggles would look like in the modified states, and then potentially still be able to, even though it's not a sequencing-based approach, it's entirely at the signal level, still be able to identify potential modifications on those strands. Okay, so I just want to summarize now where we're at and where we're headed. So in this one slide. So protein barcoding, so Lakmal mentioned protein barcoding yesterday, where they have a really powerful approach using short peptide barcodes. We're also developing a similar approach, but ours are sort of utilizing this long read approach, so we can make very, very long protein barcodes. And where we're currently at with that is prototyping, right in the middle of prototyping a library. We have around 10,000 barcodes that we estimate we'll be able to resolve against with 90% accuracy. And the cool thing about these, we are sequencing them. They're sequenceable barcodes, so they can be essentially modularized and composable. Composable is that you have individual blocks that you're able to assemble together in different sequences, is the way that you actually build these barcodes from scratch. Full-length protein fingerprinting, like I was just talking about before, where's that? How far away is that from actually going to native proteins? One, we still need to improve our sequence-to-signal models, right? So it's better data collection. Our approach requires adapters, much like the peptide-based approach, where you need these DNA handles at both ends for the short read approach. Our approach needs handles at both ends, but they're proteins, so we still have to optimize adapter chemistry to uniquely label C-terminal and the N-terminal. Approaches exist. They need to be made better and more robust so that they can be applied proteome-wide. But we are really excited about the potential of this squiggle-based approach. So what we've done is we've done these large-scale simulations in which that allows us to estimate that around half of human proteins can be identified. Full-length human proteins would be identifiable using this squiggle-based approach with about 70% accuracy, which we think would be a pretty good baseline to aim for. And then finally, what everyone really wants, a holy grail, right, is de novo protein sequencing. So we're not there yet, but I think there's an incredible amount of interesting applications for that. And I think the biggest challenge of us to get to there is really into new pore chemistries, because one thing I didn't mention, the Kamer size in these experiments when we're pulling this protein strand through the pore is close to 20 amino acids. For DNA, it's around 5, so it's an incredibly large sequence space that we have to resolve against. And if we can shrink that down and shrink that Kamer size, I think that would be going to be the quickest approach for us to make progress towards de novo sequencing, along with all these other things that we still need to do as well. Okay. And with that, I'd like to acknowledge the really great team that actually did all of this work, the funding sources, and acknowledge my conflict of interest here, having sponsored research with Oxford Nanopore, as well as a long-term collaborator and consultant with them as well. And I'll thank all of you for your attention, and happy to take questions.